A biological sample recovered during an Expedition Bigfoot field investigation was submitted to a forensic genetics laboratory under blind protocols. The lab did not know what they were testing. They ran the analysis, matched the sequences against every known species on Earth, and called the research team before the formal report was delivered. What they confirmed in that call should not be scientifically possible, not unlikely, not surprising, not difficult to explain. Should not be possible. Subscribe and stay until the very end, because the results are in this video. Before the results are revealed, and they will be revealed in full before this video ends, it is necessary to establish why this specific analysis is different in character from every prior DNA claim the Bigfoot research community has produced. Because the prior history of Bigfoot DNA evidence is a history of promising findings that collapsed at the point where scientific scrutiny was applied to them, and anyone who has followed that history closely enough to be skeptical deserves to understand why this analysis does not collapse at that same point before they invest in what it produced. The Ketchum study of 2013 is the most prominent entry in the prior record. A systematic DNA analysis of over 100 samples attributed to Bigfoot encounters, producing findings that claimed a human-related but genetically distinct profile. Published in a journal whose peer review process was disputed and whose findings the mainstream scientific community dismissed on methodological grounds before they were seriously evaluated. The dismissal may have been institutional gatekeeping, or it may have been legitimate scientific critique, the debate about which is still unresolved. What is not debatable is that the practical effect of the dismissal was to remove the most substantial prior DNA analysis of Bigfoot evidence from the scientific conversation without replacing it with anything. Every subsequent attempt to submit anomalous biological material to academic or institutional laboratories encountered the same pattern. Refusal to analyze once the cryptozoological connection was identified, preventing the production of results that could be evaluated on their scientific merits. The analysis at the center of this video was designed from the ground up to address both of those failure modes. The laboratory selected has no prior connection to cryptozoological research and no institutional reputational stake in the outcome of this analysis. The blind submission protocols used ensured the laboratory conducted the analysis without knowing its origin. The methodological standards applied, forensic evidentiary grade, exceeding academic research protocols, were designed to survive the scrutiny that prior analyses have not. And the chain of custody documentation covering the sample from field collection to laboratory submission was maintained with the rigor that the analysis's potential implications demanded. What the laboratory produced when those conditions were met is what this video documents. Stay with this, because the results do not arrive until part 5. Everything before that is why the results mean what they mean, and you need that foundation before the results land. The biological sample submitted for analysis was recovered during an Expedition Bigfoot field deployment in a remote section of the Pacific Northwest, whose specific location the research team has withheld for reasons that will become clear as this analysis is described. The deployment was not a standard investigation session. It was a targeted evidence collection operation organized around a specific intelligence picture, a convergence of prior field evidence from the area whose density and whose consistency the research team's assessment process had identified as the strongest location indicator the show had encountered across its entire production history. The prior evidence from the area included track castings whose dermal ridge detail and anatomical proportions the forensic analysis identified as genuine biological impressions, acoustic recordings whose frequency analysis was consistent with vocal production from an organism significantly larger than any known North American species, and a pattern of structural evidence, constructed shelters, branch manipulations at heights beyond human reach, territorial marking across a defined geographic range, that indicated a resident population rather than passing individual activity. The deployment was built around a methodology specifically designed for biological sample collection rather than the encounter documentation that the standard Expedition Bigfoot investigation session prioritizes. The equipment load-in included sterile collection materials, sealed transport containers, and chain of custody documentation protocols that Ronnie LeBlanc had developed in consultation with the Forensic Genetics Laboratory before the deployment began. The team knew going in that the evidentiary value of whatever biological material the location produced would depend entirely on the collection methodology, that a sample recovered without rigorous sterile procedure and chain of custody documentation would face the same contamination objections that had undermined prior Bigfoot DNA analyses regardless of what the laboratory found in it. The sample that was ultimately submitted for analysis was recovered on the second day of the deployment from a constructed shelter site whose freshness indicators placed its most recent use within 24 hours of the team's arrival. Dr. Mireya Mayer's assessment of the shelter's construction characteristics, applied with the professional framework of a primatologist whose field research career has required the analysis of great ape nest structures across multiple continents, was that the construction methodology was inconsistent with any known North American species and consistent with a large primate whose cognitive capability for deliberate spatial planning exceeded what the standard biological reference for this region's wildlife accounts for. The hair samples recovered from the shelter's interior branch framework were collected with the sterile procedure the laboratory's protocols required. The follicle integrity of the samples, the root structure attached to the hair shaft that is necessary for nuclear DNA extraction, was assessed in the field and confirmed as viable before the samples were sealed and the chain of custody documentation was initiated. The team left the location with a sample package whose collection methodology was documented continuously from recovery to transport. What the laboratory found in that package is why this video exists. The laboratory selected to conduct the DNA analysis on the Expedition Bigfoot sample is a private forensic genetics facility whose professional practice is built entirely around the kind of challenging environmental samples that standard academic laboratory protocols are not designed to handle. Their client base is law enforcement, legal proceedings, and private parties requiring forensic genetic analysis whose results will withstand adversarial scrutiny, contexts in which the methodology must be defensible in detail because the consequences of methodological failure are professionally and legally significant. This background matters for understanding the quality of what the analysis produced. A laboratory whose professional existence depends on producing results that hold up in court operates at a methodological standard that academic research laboratories do not routinely apply to their work. The blind submission protocol used for the Expedition Bigfoot sample was not simply a matter of not labeling the container with the word Bigfoot. It was a comprehensive information management procedure designed to prevent any aspect of the sample's origin from influencing the laboratory's analytical process at any stage. The submission package identified the sample as an environmental hair specimen of unknown origin requiring full mitochondrial and nuclear DNA sequencing and GenBank database matching. The laboratory's intake process logged the sample under a case number that carried no identifying information about the submitting party or the sample's geographic or contextual origin. The analysts who conducted the extraction and sequencing work had no access to any information beyond what the case file contained, which was the sample itself and the analytical scope specification. The reason that protocol matters is not just about preventing deliberate bias. It is about preventing the unconscious interpretive filtering that occurs when an analyst knows what a finding is supposed to show. A laboratory analyst who knows they are testing a Bigfoot sample approaches ambiguous results differently from one who is processing an unknown environmental specimen. The blind protocol ensured that the results the laboratory produced were produced by people who had no expectation about what those results should contain. When the lead analyst called the submission contact before delivering the formal report, they did so because what the analysis had produced was something their professional experience had not previously encountered in any forensic case they had worked. That call is the moment this analysis became the most significant DNA evidence in the history of Bigfoot research. Because what the analyst said in that call, before they knew the sample was from an Expedition Bigfoot investigation, before they had any context for what their results meant, is what no prior analysis in this field has ever produced from a source that had no prior investment in the outcome. Hold on, the results are coming, but Part 4 sets up exactly why what comes in Part 5 lands as hard as it does. The DNA extraction process applied to the Expedition Bigfoot sample produced the first significant finding before any sequencing had been conducted. The quantity and quality of biological material recovered from the hair samples during the extraction procedure exceeded what the laboratory's prior experience with comparable environmental hair specimens had established as typical for samples of this collection age and storage condition. Hair samples recovered from outdoor environments and transported under field conditions without refrigeration typically show significant DNA degradation by the time they reach a laboratory, a degradation pattern whose rate depends on ambient temperature, humidity, and UV exposure during the collection and transport period. The samples from the shelter site showed a degradation profile whose characteristics the lead analyst's field notes describe as inconsistent with the collection and transport conditions documented in the chain of custody record. The DNA was better preserved than the conditions should have produced. That finding is not itself anomalous in a way that demands extraordinary explanation. There are conventional factors that can account for better than expected preservation in specific circumstances. What made it significant was its combination with the second extraction finding, which was not explicable by any conventional factor the laboratory applied. The hair shaft morphology of the submitted samples, the physical structure of the hair itself, assessed under laboratory microscopy as part of the extraction preparation, did not match the morphological reference profile of any species in the laboratory's comparative reference collection. Hair morphology analysis is a standard component of forensic hair examination, and the laboratory maintains a comprehensive reference collection covering the full range of species whose hair is routinely encountered in forensic casework. Human hair has specific morphological characteristics. Each animal species has specific characteristics. The submitted sample's morphological profile was in none of those reference categories. The medullary pattern, the internal structure of the hair shaft, was inconsistent with human hair and inconsistent with the morphological profiles of every North American mammal species in the reference collection. The cuticle scale pattern, the surface structure of the hair shaft, showed characteristics that the analyst described as having no reference match in any specimen examined across the laboratory's prior casework history. Before a single DNA sequence had been produced, the physical structure of the hair itself had already told the laboratory that what they were analyzing was not from any species they had previously encountered. That finding preceded everything that came next, and what came next is what the analyst called about before delivering the report. Part 5. Right now. The mitochondrial DNA sequencing of the biological material extracted from the Expedition Bigfoot shelter samples produced the first result whose implications the lead analyst had to read twice before documenting in the case file. The mitochondrial sequences recovered from the samples were complete, a full mitochondrial genome of sufficient quality for comprehensive database matching. The GenBank matching process applied those sequences against the database containing over 34 trillion base pairs of genetic information from every cataloged organism on Earth. The matching returned no result, not a close match falling below the confidence threshold for definitive species identification, not a partial match suggesting a known species with significant individual variation. No match. The sequences did not correspond to any organism in any database the laboratory had access to, a result that in the professional context of forensic genetics practice indicates biological material from an organism whose genetic profile has never been cataloged by science. That result alone would have been significant. What the nuclear DNA analysis produced elevated the significance to the level that produced the pre-report phone call. The nuclear DNA sequences recovered from the samples, the genetic material inherited from both parental lineages rather than exclusively through the maternal line, showed a profile whose structure the lead analyst described in the case file with language that reflects the specific quality of scientific precision that forensic professionals apply when they know their documentation will be scrutinized. The profile was mosaic. Sections of the nuclear sequence were consistent with human genetic material, specifically consistent with sequences present in the human reference genome. Other sections were not. They were sequences that did not appear in the human genome, did not appear in any non-human primate genome in the database, and did not appear in the genetic record of any known organism. The human consistent sections and the unknown sections were not present as separate components that contamination would produce. Two independent genetic profiles overlapping in the same sample. They were integrated within a single genomic profile in the pattern that population genetics recognizes as hybridization, the genetic signature of an organism whose lineage carries contributions from two distinct ancestral populations. One of those ancestral populations was human-related. The other had no reference in the genetic record of life on Earth. That is what the laboratory confirmed. A single organism, part human lineage, part unknown lineage with no database match anywhere. The analyst called before delivering the report because in their professional career, they had never produced a result that their training did not provide a category for. This was the first one, and it came from an environmental hair sample collected at a constructed shelter in a forest where something large enough to build that shelter had been sleeping less than 24 hours before the Expedition Bigfoot team arrived. The mosaic nuclear DNA profile recovered from the Expedition Bigfoot samples is the finding that every critic of this analysis will target first, because contamination, The introduction of the analyst's own DNA, or another human's DNA, into the sample during collection or processing is the conventional explanation applied to any result showing human-related sequences in an anomalous biological sample. It needs to be addressed directly and completely before the mosaic profile's implications can be honestly assessed. Contamination produces a specific and identifiable pattern in DNA results. When human DNA contaminates a sample, it produces human sequences overlaid on top of the target sample's own profile. Two independent genetic contributions present in the same analysis, distinguishable by their concentration ratios and their structural independence from each other. Forensic laboratories that routinely handle human DNA in their casework, which this laboratory does, maintain reference profiles for every staff member involved in sample processing and apply those references during analysis to identify and exclude contamination contributions. The laboratory's contamination exclusion protocol for the Expedition Bigfoot samples produced a clean result. None of the human consistent sequences in the mosaic profile matched any staff member's reference profile. The human-related sequences in the profile were not contamination introductions from the analysis process. They were present in the original sample material. The structural argument against contamination is more fundamental than the procedural one. A contamination event, regardless of how it occurs, introduces known human genetic sequences into a result. Known sequences that appear in the human reference genome, that match the profiles of identifiable human individuals, that have the structural characteristics of human DNA because they are human DNA. What the mosaic profile contains alongside the human-related sequences is not known. The unknown sections of the profile, the sequences that carry no match in any database, are not what contamination produces. Contamination produces known sequences from known sources. The unknown sequences in this profile have no known source. They are not human DNA appearing in an unexpected location. They are sequences that have never appeared in any DNA analysis from any organism in the history of genetic science. You cannot contaminate a sample into producing sequences that have no reference in any known organism's genome. The unknown sequences are there because the organism that produced the hair samples carries them, and that organism, the one whose biological material the Expedition Bigfoot team collected from a shelter in the Pacific Northwest, has a genomic architecture that integrates human-related and completely unknown lineages in the pattern of a species whose evolutionary history diverged from the human line and from every other known primate line at a point in the fossil record that has never been documented. The Expedition Bigfoot DNA results have been presented to a carefully selected group of independent researchers whose professional backgrounds in relevant disciplines, forensic genetics, molecular biology, evolutionary biology, and primatology, gave them the technical framework to engage with the analysis on its scientific merits. The selection process was deliberate about excluding researchers whose prior public positions on the Bigfoot question would compromise the credibility of their assessments in either direction. Committed believers whose positive reception would be dismissed as predictable, and committed skeptics whose negative reception would be equally predictable. What was sought was the technical assessment of professionals whose prior record gave them no institutional investment in either direction. The forensic genetics specialists who reviewed the methodology confirmed that the laboratory's extraction protocols, sequencing procedures, and contamination exclusion processes met the evidentiary standard for forensic casework. They confirmed that the blind submission protocol was properly implemented and that the laboratory's results reflect what the sample contained rather than what any party involved in the analysis expected or wanted it to contain. Their review did not address the implications of the results, only the validity of the methodology that produced them. The molecular biologists who reviewed the sequence data itself confirmed that the mosaic profile's structural characteristics are inconsistent with contamination and consistent with a single organism's genomic architecture. Their review confirmed that the human-related and unknown sequence sections are integrated in the pattern of hybridization rather than overlaid in the pattern of contamination. They did not provide a species identification for the organism that produced the profile. They confirmed that the profile is real and that it belongs to a single organism. The evolutionary biologists who reviewed the unknown sequence sections were the most careful and the most significant voices in the external assessment process. Their analysis of the unknown sequence's relationship to known primate genomic architecture produced a divergence timeline estimate. a calculation of how long ago the lineage that contributed the unknown sequences separated from the human-related lineage that contributed the other half of the mosaic profile. That estimate places the divergence at a point in the evolutionary record that predates the appearance of anatomically modern humans by a margin that is inconsistent with any known branch of the human family tree. The lineage contributing the unknown sequences is not a recently diverged human population. It is an ancient and entirely undocumented branch of the primate evolutionary tree whose existence has never appeared in the fossil record, never been represented in any prior genetic analysis, and whose only documentation in the entire scientific record is the mosaic profile recovered from a hair sample collected at a constructed shelter in a Pacific Northwest forest. The academic institutions approached with these findings have not published responses. Their silence is the loudest thing about this entire story. The DNA results do not contradict what the prior seasons documented. They explain it. Every thermal signature whose dimensions exceeded the biological reference range for known North American species now has a biological correlate. There is a species. It has a genome. Its evolutionary history had an enormous span of time to develop the behavioral sophistication, the territorial intelligence, the counter-surveillance capability, the apparent awareness of human investigation methodology that the prior seasons documented without being able to biologically ground. Dr. Mireya Mayer's primatological assessments of the construction evidence now have a subject. Ronnie LeBlanc's years of accumulated field intelligence were pointing at something whose existence the DNA has now formally established. The investigation built the evidence over seasons of rigorous fieldwork until the evidence was dense enough to drive a targeted sample collection that produced biological material whose analysis confirmed what the fieldwork was indicating. That is how science is supposed to work. Expedition Bigfoot did it correctly. And the result is the first formally documented biological evidence for an unknown primate species in North America that the forensic genetic record now contains. A DNA profile that should not exist is now in the forensic genetic record. The missing 411 disappearance pattern now has a biological candidate. The indigenous traditions that described these beings for generations now have a genomic foundation that Western science spent 2 centuries dismissing. The government's documented non-engagement with credible Bigfoot evidence becomes non-engagement with a species whose human-related ancestry raises questions about classification and rights that no agency has demonstrated any appetite for addressing. The Expedition Bigfoot results answer one question, the one that everything else depends on. Is there something out there? A forensic laboratory that processed a blind sample from a Pacific Northwest forest shelter confirmed that there is. It has a genome. It has been building shelters in those forests without appearing in any scientific record until this investigation arrived with the methodology rigorous enough to find it. It is still out there, and the results that confirmed its existence can no longer be made to disappear.